Assalamu alaikum, dear student. This is your biology class, and I'm your mentor, Sir Humayun Ali, and we will continue our topic 8.2, Part 2, Calvin cycle, from unit number 8, Bioenergetics, and the role of chlorophyll in photosynthesis. Now, we will start our topic with the discovery of a Dart reaction, which is also named as Calvin cycle, discovered by scientist Melvin Calvin. And please note that this reaction takes place in the thylakoid of the chloroplast. The Dart reaction or Calvin cycle has 3 steps. The first one is carbon fixation, the second one is reduction, and the third one is regeneration. We will start our first step of Dart reaction, which is carbon fixation. Here you can see the 3 molecules of carbon dioxide combined with the 3 molecules of 5 carbon molecule, which is already present in the stroma of the chloroplast, and they form a temporary unstable 3 molecule of 6 carbon compound, which will split into 6 molecules of 3 carbon. Here comes the second step, which is reduction. Out of the previous 6 molecules, 1 molecule of 3 carbon get reduced by NADPH by using energy from the ATP and form a 3 carbon sugar. And please note that this is the 3 carbon sugar, and the glucose which we are going to form is 6 carbon molecule. So this process or step or the cycle continue 2 times. Remember that the Calvin cycle continues 2 times to form 1 molecule of glucose. Now, this is the last step of Calvin cycle, which is regeneration. That meaning 5 molecules of 3 carbon rearrange themselves, and with the help of ATP, by using the energy from the ATP, they form again 3 molecules of 5 carbon compounds, which ultimately again react to combine with the carbon dioxide and start the process. Again, for your clarity, I will summarize the Calvin cycle in this single slide. The Calvin cycle is comprised of 3 steps. The first step is the carbon fixation, in which carbon dioxide is attached with the 5 carbon molecule, which is already present in the chloroplast, which is highly unstable and split into 6 molecules of 3 carbon, out of which 1 gets reduced and form 3 carbon sugar, as you can see in the second step, that is reduction. And then in the last step, the remaining 5 molecules of 3 carbon rearrange themselves and form the 5 carbon molecules again, and this cycle continues 2 times to form 1 molecule of glucose. And now our next topic is a role of chlorophyll in photosynthesis. There is a term that is present in the side box. The term is pigment. What are basically pigment? Pigments are the substances that capture different wavelength of sunlight. Basically, these are the molecules and they are of various types, and they capture different wavelength of the sunlight. Each capture a specific wavelength of the sunlight, and these substances are called pigments. And there are various types of pigments, such as chlorophyll A, which is the main photosynthetic pigment, which absorb mostly a red and blue wavelength of the sunlight, and the other are called accessory pigments, such as chlorophyll B and carotenoids, etc. This slide will tell us which type of wavelengths are absorbed by the different types of pigments, such as chlorophyll A mostly absorb red and blue wavelength, which also comes in the board in the form of MCQs, and the yellow and orange and green are absorbed by other pigments such as chlorophyll B and carotenoids. And as you can see in your last slide that only the 1% of the sunlight is captured by the leaf of the plant. That's 99 is reflected or transmitted into the atmosphere. And this part also comes in MCQs.